In eukaryotic organisms, there is a very important step that precedes the translation of the mRNA code into polypeptides and RNA that our genes code for. This step occurs during the processing stage of the pre-mRNA that was transcribed from the template DNA strand in order to prepare it for export out of the nuclear envelope and into the cytoplasm. This step is called RNA splicing. This is our strand of pre-mRNA. These are the 5' and 3' ends. To simplify our example, we only have one intron, the sequence in red, and 2 exons, which are both yellow. Every intron starts with the code GU and ends with the code AG. The phosphodiesterase bonds that connects the beginning of an intron to the first exon is our 5' splice site, and similarly, the phosphodiesterase bond between the end of the intron and second exon is the 3' splice site. In between the splice sites lies an adenine, which is called the branch point. Lastly, jutting off from that adenine is an OH group with a special function in RNA splicing. This is our pre-mRNA. Splicing is facilitated by a complex of proteins and RNA called a spliceosome. Within the spliceosome are 6 snRNPs, each with their own function on RNA splicing. First, the U1 protein identifies the correct intron by binding complementary to the RNA near the GU start site. Shortly thereafter, the U2 SNRP comes in and also binds to the GU site and causes the RNA to fold to the branch point. U5 and U6 also come in and complete the spliceosome. Then a reaction called transesterification is triggered by the phosphorylation of the U5 SNRP, which causes the OH group on the adenine in the branch point to attack and break the bond between the intron's guanine and the exon sequence. This opens up a carbon on the guanine so that it can bond to the adenine, forming an unusual 2' 5' phosphodiester bond. This loop structure is now called a lariat. The released OH then binds to the end of exon 1.
Next, another transesterification reaction occurs between the end of exon 1 and the start of exon 2. The U5 SNRP brings the 2 exons in close proximity, and after being phosphorylated again by an ATP, the OH attacks the bond at the 3' splice site. This breaks the intron sequence away from the exon sequence, and the 2 exons are covalently bonded together. The spliced intron is then released from the SNRPs, the spliceosome dissociates, and the intron is recycled into nucleotides. Because it allows for a single gene to be spliced in various ways and code for multiple polypeptides or RNA products, alternative splicing increases our genetic diversity. However, we don't know what decides how or where a pre-mRNA is spliced, but according to Ms. Rankin, if you do, you will win the next Nobel Prize. Like all genetic processes, no one mechanism is free of mistakes, and splicing is no exception. Because a spliceosome recognizes certain patterns such as GU and AG, if a mutation in either the DNA or pre-mRNA codes change, the spliceosome may cut the sequence in the wrong place. In fact, more than 15% of all human diseases are caused by splicing errors. This is most commonly seen at branch points. The mutation then leads to incorrect splicing, which interferes with the lariat formation. One such example of a disease that can result from incorrect splicing is cystic fibrosis. In the end, we hope to learn more about what decides splicing and how we can use this knowledge to identify mutations that are at risk for incorrect splicing to ultimately prevent them.